Welcome, everyone. Claret Bio's mission is to develop tools to generate high-quality next-generation sequencing data from challenging samples such as liquid biopsies, FFPE, rootless hair, and ancient DNA. Our flagship library preparation method seriously works very well with such input types. Given the gradual adoption of Seriously in various labs, we decided to conduct a webinar series to showcase the research of our customers and collaborators and discuss the challenges and solution to generating high quality NGS data. Of course, having the first author of a paper present the data is probably more interesting than doing a simple journal club. To kick off this series, we have our first speaker today, Ms. Adrienne Chang, who will present her work from her recent paper in clinical chemistry that used Seriously. She's a PhD candidate in biophysics at Cornell University. She obtained her bachelor's degree in chemistry from New York University, Abu Dhabi. Her research under the mentorship of Dr. Ivan D. Vlamnick focuses on developing tools for infection and immune response profiling. Her work evaluates the biases introduced in metagenomic sequencing assays and their effects on the sensitivity of liquid biopsies for tuberculosis diagnostics. Her title for today's talk is Methods and Applications of Metagenomic Cell-Free DNA Sequencing Assays. Welcome once again, Adrienne.
Thanks for the really kind introduction, Barsha. As you mentioned, today I'll be talking about methods and applications of metagenomic cell-free DNA sequencing assays. So there are more than 1,000 microorganisms that are known to cause human disease. And despite the many interventions available on the market, infectious and microbiome-associated diseases remain a leading cause of death worldwide. Differential diagnosis of disease is complex and it's a challenging task in the clinic. So conventional diagnostics, like culture, are not sensitive to co-infection or the detection of unculturable microorganisms. Next generation sequencing, though, offers an unbiased and rapid screening for a broad range of potential pathogens. A typical NGS workflow has three major experimental steps. So in the first step, we isolate DNA from a biofluid sample, and this can be plasma, urine, or many other different sample types. From there, we can prepare DNA sequencing libraries from the isolated DNA. And finally, we can, of course, sequence these DNA libraries. In our group, we are interested in looking at the potential of cell-free DNA as an analyte for infectious disease diagnostics. So cell-free DNA is highly degraded DNA that originates from human, bacterial, and viral cells, and it's ubiquitous in biofluids. It primarily derives from apoptotic cells, and thus it can serve as a way to measure cell turnover and death. There are already a number of clinical applications for cell-free DNA, including in non-invasive prenatal testing, where you can detect fetal DNA, in cancer screening, looking at tumor-derived cell-free DNA, to monitor transplant rejection by looking at donor-derived cell-free DNA, and of course, in infectious disease diagnostics. There's many challenges to developing a robust cell-free DNA metagenomic assay. In my talk today, I'll just cover a few of them and provide some solutions for how we've tried to address them. So the first question is, how can we capture the diversity of cell-free DNA fragments, which are known to be degraded? Conventional DNA library preparation relies on double-stranded adapter ligation on the left, where double-stranded adapters are ligated onto the ends of DNA. And using bead-based size selection and PCR, we eventually end up with a DNA library. In 2016, Phil Burnham, a graduate of the lab, had a new idea that was inspired by genomic analyses of ancient DNA. And he proposed a single-stranded DNA library preparation method. In this case, the first step is actually to denature all the DNA using high temperature, separating all the DNA in a sample to single-stranded DNA. We can adapt single-stranded DNA ligators onto the ends of the DNA, extend the DNA, and then we follow pretty much the same as conventional double-stranded DNA library preparation. We applied this method and conventional library preparation to 40 matched plasma samples and looked at the length profile. We can first look to see at the length profile for chromosomal and mitochondrial DNA when we use conventional double-stranded library preparation. And we find a very narrow range of fragments that conventional library preparation is sensitive to. So really only around 100 to 200 base pairs in length. We find with single-stranded library preparations, we are much more sensitive to the full range of cell-free DNA length. We're finding a large majority of peaks that are ultra-short or less than 100 base pairs. And this is especially so in the case of mitochondrial cell-free DNA. So using this single-stranded library preparation, we can look at the biophysical properties of cell-free DNA. So first on the left, looking at chromosomal cell-free DNA, we can look at the fragment length profiles of plasma in red and urine in blue. And we can see that plasma is on average a lot longer than urinary cell-free DNA. And it exhibits this peak at around 167 base pairs, which we know corresponds to the length of DNA wrapping around histones, which protects it from nuclease degradation. We can also look to see that the fragment length profiles of plasma and urinary cell-free DNA that are part of the mitochondrial and microbial cell-free DNA subsets are both ultra short, with an average length of less than 100 base pairs. What was really exciting to see was that using the single-stranded DNA library preparation, we were able to have more efficient recovery of microbial sequences. So in these matched samples, we quantified the genome equivalence relative to the coverage of the human genome for bacterial, eukaryotic, and viral genomes. On the y-axis is the relative genome equivalence using this double-stranded DNA method, and on the x-axis is the single-stranded DNA library preparation method. And we find that we're getting a 71-fold mean increase in microbial sequences. We can also look specifically just at the bacterial fraction, which makes up around 89% of the species sampled. And we are finding a 74-fold increase in bacterial reads when we use the single-stranded DNA library preparation protocol. And so we're finding that the choice of a library preparation has a significant effect on the length profile measurement and the recovery of cell-free DNA. And this got us thinking, because microbial cell-free DNA constitutes maybe 1% of the total reads that we sequence. And so the relative recovery of microbial cell-free DNA compared to the host DNA affects the sensitivity of a metagenomic cell-free DNA sequencing assay. And so the next question we want to address is understanding how the sample preparation workflow affects the sensitivity of a metagenomic cell-free DNA sequencing assay. And so this is the paper that Varsha introduced earlier that was recently published in Clinical Chemistry, where we looked at assay-specific biases in the sample preparation workflow for cell-free DNA studies. So for the first part, we created measurement bias models where we took phi-x-DNA that was sheared to 5 different target lengths and then mixed so that we had a fragment length profile that was broad and smooth, ranging from 0 to 500 base pairs. And we took this mixture and we subjected it to either cell-free DNA isolation or library preparation. Before and after each of these assays, we quantified the assay by using the fragment analyzer and a qubit measurement. And using these assays, we could then look at the efficiency of an assay by taking the abundance and fragment length before and after the assay to describe a transfer function for each assay. And we did this for 5 different DNA isolation kits and 3 methods of library preparation. And we can see right off the bat that there are differences in efficiency for both the DNA isolation kits and library preparation protocols. So for example, 3 of these kits, the CNA, MagMax, and modified MagMax, shown in blue, pink, and purple, were relatively insensitive to the fragment length of our input. Whereas kits like the Norgen and QIAQuick in yellow and green had some strong biases where they were highly efficient at obtaining short fragments and maybe less so as the fragment length increased. We also see that for library preparation, our 2 single-stranded protocols, the Meyer, which I introduced previously, and the Seriously kit, also had some relationship between the fragment length and the efficiency of recovery, whereas the NEB, which was our double-stranded library preparation, was relatively insensitive. And so for each overall sample preparation, we can describe an overall transfer function by taking the product of the efficiency curves for cell-free DNA isolation, library preparation, and sequencing. And an example of what that combined transfer function looks like is shown here on the right. Using this model, we assayed 305 urine samples using 5 different cell-free DNA isolation kits, and we looked at their fragment length profiles. Again, for chromosomal cell-free DNA and mitochondrial DNA, we are seeing differences based on the cell-free DNA isolation kit used. So that Norgen kit that was highly efficient at recovering short fragments, again, we're seeing a strong peak here near the shorter fragments than say the CNA kit, which did not favor short fragments as strongly. For microbial cell-free DNA, we are already seeing that even though we're using a different kit, the fragment length profile is really similar. And this suggests that the distribution of lengths for microbial cell-free DNA is already very narrow. And so it's not as sensitive to distortions in fragment lengths. We can then take the appropriate transfer functions and apply it to our measured density to compute an intrinsic density profile. And what we can see is that for all the kits, all 5 kits, and for all 3 types of cell-free DNA, chromosomal, mitochondrial, and microbial, we're starting to see one single fragment length profile. Not only that, we're observing a few other things. We are finding that around 89% of chromosomal cell-free DNA fragments and around 92% of mitochondrial cell-free DNA fragments are actually less than 100 base pairs. We can look also just at an example of urinary cell-free DNA. And so the measured profile is still short, but of course with this intrinsic profile, we are seeing now this very strong peak that's less than 100 base pairs. But what's exciting is that we're able to reveal this nucleosome peak at 167 base pairs. We did the same by taking these transfer functions and applying it to plasma cell-free DNA sample. And while the distribution doesn't change as dramatically as urinary cell-free DNA, we are again seeing an increase in the amount of short fragments that's intrinsic to plasma cell-free DNA. And we're able to start to recover this dinucleosome peak at around 300 or 308 base pairs. And so given that microbial cell-free DNA is ultra short and that different cell-free DNA isolation kits and library preparation strategies perform differently at different fragment lengths, we wanted to see if microbial enrichment was strongly correlated with the recovery of short cell-free DNA fragments. To do this, we created a model where we took the intrinsic density, multiplied that by the efficiency of the sample preparation workflow or the transfer function. And by taking the area under the curve of the resulting fragment length profile, we could compute the expected yield of the assay. We find that for cell-free DNA isolation kits, our models do predict the enrichment of microbial reads. So the blue diamonds show the expected enrichment relative to the QIAGEN circulating nucleic acid kit. And we can see that the Norgen kit and the QIAQuick kit, both of which that had better efficiency of recovery for short fragments, also had greater microbial enrichment. In fact, for the Northern kit, we found on average a 3-fold enrichment of reads. However, for the library preparation protocol, we did not see what we had expected. So based on our models, we would expect that the NAB double-stranded library preparation kit would result in greater microbial enrichment, while the seriously kit would have lower enrichment. But the opposite turns out to be true. The seriously kit performs significantly better than the NAB kit at recovering microbial reads. And this, we believe, is just a result of the physical configuration of cell-free DNA, which is ultra short, it's highly degraded, and it's insensitive for the double-stranded protocols are insensitive to capturing all these fragments. They're really ideally suited for fragments that are blunt-ended, such as our measurement models made with cfDNA. And so we show that the sensitivity of a metagenomic cell-free DNA sequencing assay is influenced by the DNA isolation strategies. So we can easily improve them by using strategies that favor short fragment recovery. And what's more important is the use of a single-stranded library preparation to be able to capture all of the cell-free DNA fragments that are in our samples. Another major problem for cell-free DNA metagenomic assays is environmental DNA contamination. It's a problem in most DNA sequencing assays and is pretty much unavoidable. But for low microbial biomass samples, like our cell-free DNA samples, contaminant DNA can easily overwhelm the signal. So contamination can come from a number of places, commonly your skin, from the labware you use, or the chemical reagents. And it can really confound the results that we are sharing. So in 2014, there was a paper that came out in Microbial claiming that the placenta harbors a unique microbiome. And unfortunately, subsequent papers showed that this microbiome was actually due to contamination. Contamination coming from the kitome, so the reagents we use, the splashome, or just the lack of controls. And there's currently no way of removing contaminants. Commonly, they recommend to work in a clean room facility and to use the use of negative controls. There are also a number of batch correction algorithms to try to correct for contamination after samples have been sequenced. And these usually rely on detection of any species that are found in the negative controls, and then using subtractive filtering to remove those from the sample. This runs into a lot of problems, and one of them is that there are a lot of species that are both DNA contaminants and also infection causing. And so a major challenge we wanted to address is how we can contend with environmental DNA contamination in low biomass studies. So we've developed recently a metagenomic DNA sequencing assay that is robust against environmental DNA contamination. We call this cfSeq, which stands for Sample Intrinsic Microbial DNA found by tagging and sequencing. And this was developed in conjunction with 2 other graduate students in the lab, Omari and Alex. How it works is we chemically tag sample intrinsic DNA using any kind of chemical tag that can be recorded via sequencing. After that, any type of manipulation in the sample preparation workflow that introduces contaminant DNA does not have that chemical tag. And we've developed a bioinformatic workflow to accompany this. For the practical implementation of cfSeq, we are using chemical tagging of DNA using bisulfite salt. So bisulfite salt converts unmethylated cytosines to uracils. And then in subsequent DNA synthesis steps that are part of the library preparation protocol, these are eventually converted into thymines. And so any contaminant DNA that's introduced after we do the chemical tagging can be identified by the lack of cytosine conversion. However, bisulfite treatment is extremely harsh and it increases DNA degradation. So here I'm showing a fragment length profiles again for chromosomal, mitochondrial, and microbial DNA. And we can see that the bisulfite treatment in red, again, increases the fraction of fragments that are less than 100 base pairs. So for these, it's especially important to have a preparation protocol that is able to capture these short fragments and sequence them. And so for the studies, we use the seriously library preparation protocol in combination with 2 of the cell-free DNA isolation kits that favor short fragments. We first did 2 proof of principle experiments using cDNA, which was sheared and at different concentrations. In this first experiment, we performed standard sequencing as well as cfSeq. And then we quantified the abundance of Cutibacterium acnes, which is a common skin contaminant. We find that for standard sequencing in green, as we expected, as the input biomass decreases, the abundance of Cutibacterium acnes that we detect increases. However, for cfSeq, we are not seeing such a correlation. In the second experiment, we wanted to evaluate the bioinformatic filtering steps. And so again, using sheared cDNA, we spiked in 1 nanogram of a well-characterized microbial community. Again, on the top, we quantified the abundance using conventional sequencing. And we see that as the library preparation input decreases, the abundance of all of these microbial species increases. And again, for cfSeq, we are not seeing that. We can quantify the total abundance of this microbial community. And we see that after filtering, we are resulting in a 99.8% decrease in the number of molecules that map to the microbial community. So we are significantly accounting for environmental DNA contamination. We applied cfSeq to 196 samples from 6 independent study cohorts. And we quantified the abundance of 68 genera that were identified as the most common contaminants in literature. And here I'm just showing a heat map. That's the abundance of the top 25 of the 68 that we're seeing in our sample sets. And for each of these cohorts, we see a pair of heat maps where on the left is before filtering and on the right is after cfSeq filtering. And we're seeing that across all the study cohorts, we're removing a lot of the environmental DNA contamination. In fact, 77% of the 68 genera were completely removed from all the samples after filtering. And then in the remaining genera, we are seeing up to 3-fold reduction in the number of molecules originating from the contaminant genera. We again quantified the abundance of Cutibacterium acnes, and we first found it in all of the samples before filtering. After filtering, we're removing C. acnes completely from 50 of the samples. And in the remaining samples, we find up to a 2-fold reduction in the number of breeds that align to Cutibacterium acnes. We also wanted to evaluate how well cfSeq works in removing batch effects. And so to do this, we calculated the Bray-Curtis dissimilarity index for all of our samples. And so an index value of 0 indicates that 2 species are highly similar, and 1 indicates that 2 samples are highly dissimilar. And we grouped all our samples by the sequencing run, by the operator, by the cohort, whether or not it had a urine culture, and the biofluid. And we can see that before filtering, we have these clusters that correspond to these different variables that indicate that there are batch effects present. After filtering, we find that we're reducing the noise resulting from environmental DNA contamination by up to 3 orders of magnitude. And really the only cluster we're left with is this cluster of individuals that are from Uganda, whereas all the other samples are from the US. So this indicates true differences in the microbiome profiles. We applied cfSeq to a number of different applications, but today I'll just share a few of them. In the first, we looked to see whether or not we could use cfSeq to more accurately screen for urinary tract infections and to characterize the urine microbiome. So for this, we had 26 urine samples, a few of which had positive urine cultures. On the bottom here is the unfiltered samples, and you can see that we're specific and sensitive to all of the pathogens identified by urine culture, and these are indicated by the black stars. After filtering, we're finding that, again, we are still equally sensitive to all the identified pathogens by culture, but we're more specific to only identifying those in the sample. Furthermore, we can quantify the total microbial load, and we find that there is a significant difference in the microbial load in samples that have a urinary tract infection versus those that do not. But we were not able to see this before filtering, shown here in red. We also wanted to see how well cfSeq performs in identifying viral and bacterial co-infection. So for this, we looked at 30 plasma samples from 14 individuals that had COVID-19 infection. And for 3 of these individuals, they had positive lung or blood cultures. Again, for both, we're seeing that before filtering, shown on the bottom, we're able to detect all of the species that have been identified by blood or lung culture, again, shown with these black stars. But after filtering, we're still sensitive, but we're much more specific for these microbes. Another example is we looked in 15 plasma samples from patients in the ICU, 8 of which had sepsis and had positive blood cultures for sepsis. Again, we're seeing the same trend where we are more specific after filtering than before filtering, but still equally as sensitive to all of the pathogens identified by culture. One last application that was a kind of fun exercise was to look to see whether or not we could identify signatures of bacterial translocation from the gastrointestinal tract. And so for this, we had 44 patients seeking treatment for IBD, for which we had matched plasma and stool samples. And using metagenomic stool DNA, we then mapped our plasma cell-free DNA to the species identified. And we can see that for individuals without IBD, there are very low, if any, signatures of bacterial translocation in plasma. However, the diversity of these GI tract species increases, especially when we start to look at individuals with ulcerative colitis. For 9 of these samples, we had matched samples pre and post medical therapy. So again, we did the same exercise, except this time we wanted to see if we could identify changes in the signature of bacterial translocation due to therapy. And what we find is that after therapy, the majority of the species, we're no longer seeing these signatures. These are indicated by these downturned arrows that are filled in. And for many of the other species, we're seeing a significant reduction in the amount of bacteria in plasma after therapy. And these are shown by the outlined downturned triangles. What was interesting is that for healthy gut bacteria like lactobacillus, we're seeing the opposite trend. In fact, we're seeing an increase in the abundance of bacteria in plasma that might be a signature of bacterial translocation. And in this sample in particular, we're seeing up to 12.7-fold increase in lactobacillus after therapy. So we proposed a new method to contend with environmental DNA contamination by using cfSeq, which uses a chemical tagging method to identify contaminant DNA. But this still doesn't get to kind of the main problem of diagnostic, which is how do we separate infection from disease? So infection is not synonymous with disease. You can have microbes colonizing you, but they're not necessarily causing disease. And so maybe a better way to define infection is requiring an interaction between the host and a microbe that causes some sort of host injury. There are many ways that we could potentially quantify this host injury response. But one way that we do it in our lab is by performing methylation profiling. So every tissue and cell type has a unique methylation pattern, and we can use reference methylation atlases to describe all the tissues and cell types in our body. And we can perform bisulfite sequencing on our cell-free DNA to then compute the tissue of origin of the cell-free DNA. We've applied this to a number of different contexts, but again, today I'll just share a subset of these examples. These analyses were largely spearheaded by Alex Chang in our lab, so I hope I'll do him justice on these. In this first study, we wanted to look to see whether or not we could monitor urinary tract infection after kidney transplantation. So we performed whole genome bisulfite sequencing on urinary cell-free DNA after in individuals who had kidney transplants. These individuals were further broken down into 6 subcohorts. In the first were individuals who did not develop a UTI after a kidney transplantation. There was a group who did develop urinary tract infections, and there were a group of individuals that had infection with BK polyomavirus. Within these, we had some that had biopsies that indicated nephropathy or kidney damage, and this is the BKVN group. We also had ones that in their biopsy, they did not show any nephropathy. That's this BKV positive and minus group. And we had ones that did not have any liquid biopsies, but had signs, other clinical signs of BK polyomavirus infection. And these are the normal and early groups. And so we took samples of urine and performed whole genome bisulfite sequencing on the cell-free DNA. And from this, we can get both the tissue of origin of cell-free DNA, as well as look at the microbial composition. And so in DNA, we're just showing that the methylation patterns do indeed separate different cell and tissue types correctly. So we can see things like the small and large intestine tend to group and cluster together, whereas immune cells such as NK cells, T cells, and B cells also cluster together. And then these solid organs also cluster in their own separate way. We can quantify first the donor fraction for individuals who had sex mismatch transplants by quantifying the abundance of the Y chromosome relative to the coverage of all autosomal chromosomes. And we can see that the majority of these individuals, their cell-free DNA comes from either the kidney or urinary tract tissue or from immune cells, as we would expect. We can use this donor fraction and look to see how well it correlates with the fraction of cell-free DNA that derives from kidney tissue. And we see a very strong correlation, suggesting that there is damage to the kidney that is related to the amount of donor DNA that's being released into the blood. We can also look to see how the proportion of cell-free DNA coming from different tissues changes with different disease outcomes. So we see that for individuals that had BK polyomavirus, which can result in kidney damage, we are seeing an increase in the relative proportion of kidney-derived cell-free DNA as compared to individuals with UTI or no UTI. We can also use this information to look at temporal dynamics of cell-free DNA. So these are samples from individuals that did not develop a UTI infection, and we can look to see what their cell-free DNA abundance looked like for each tissue along their post-transplantation timeline. So we can see this pretty big spike around day 3 of transplantation, which corresponds to host injury as it's starting to heal from post-operative stress. This goes down by around day 10, and by day 100, so around 3 months after transplantation, they're around back to baseline. Again, we can quantify the amount of kidney-derived cell-free DNA, and we're finding that in individuals with BK polyomavirus with nephropathy, we're seeing a significantly increase in the amount of kidney-derived cell-free DNA. And in fact, the amount of increase is significant enough to differentiate it from individuals with BK polyomavirus, but without nephropathy. One other thing that we started to look into was to look to see how the mean fragment length changed in different populations. So we are finding that in individuals with urinary tract infections, the mean fragment length is shorter than those without urinary tract infections. And this is actually related to the abundance of leukocyte-derived cell-free DNA. And based on literature, this makes sense because the environment of leukocytes does promote DNA degradation. And so we'd be interested to look in the future at how patterns of degradation might provide an additional metric to stratify different infectious pathologies. And of course, using whole genome bisulfite sequencing, we're still able to identify clinically relevant pathogens. So in this first panel, we're just showing that the abundance of bacterial and viral genomes using whole genome bisulfite sequencing on the y-axis is the same as using standard sequencing. So we're not decreasing our sensitivity. We can again quantify the abundance of BK polyomavirus, and we're really only seeing it in these two subcohorts that have BK polyomavirus infection. In individuals that had positive urine cultures, we are also able to detect the pathogen that was identified through urine culture with high specificity and sensitivity. And finally, we can look to see how the effect of BK polyomavirus infection, bacteria infection, or co-infection affects the abundance or the tissue damage. So this cluster of individuals had a lot of individuals that had BK polyomavirus infection or BK polyomavirus and bacterial co-infection. And we find that these individuals have a significant proportion of tissue-derived cell-free DNA from the kidney. However, this cohort down here, we're not seeing that. In fact, we're just seeing an increase in the tissue proportion coming from neutrophils. And this actually corresponds to individuals that only have bacterial infections. One last example that I'll share with you all is the ability to use these kinds of assays to monitor hematopoietic cell transplantation complications. And so in this cohort, we had plasma cell-free DNA in which we can look at a number of different outcomes. And I'll just share the example of looking at tissue damage and graft versus host disease. So for individuals with blood disorders, there are 3 different time points in which we sampled plasma before hematopoietic cell transplantation, and then additional sampling after transplantation. One major complication after cell transplantation is graft versus host disease. This can be extremely fatal, and so we use cell-free DNA concentration to evaluate whether or not we could identify GVHD prior to clinical onset. And in fact, we are able to see that in someone with severe GVHD, shown on the top, we're seeing a dramatic increase in the concentration of cell-free DNA in the days post-transplantation, as compared to an individual who did not develop GVHD. We're again able to observe temporal dynamics in response to conditioning and transplantation. So here we're just looking at a subset of the cell-free DNA concentration and only the solid organ fraction. And we can see that in the 3 pre-transplant time points, there's really no difference in the solid organ fraction between GVHD positive and GVHD negative individuals. However, after transplantation, we're seeing that individuals who developed GVHD had significantly higher concentration of solid organ-derived cell-free DNA. And we can look more specifically here just at the days post-transplant and see that for individuals who did not develop GVHD, the solid organ concentration resolves to baseline much faster than those with GVHD. And so I've presented now a number of different ways we've addressed some of the challenges that we face in developing a robust cell-free DNA metagenomic assay. So in the first, in order to capture cell-free DNA fragments, it's really important to think about the library preparation protocol you use. Different protocols are sensitive to different types of fragments. And for cell-free DNA, which can exist in many different degraded states, single-stranded library preparations really outperform conventional double-stranded library preparations. And not only that, at least for metagenomic cell-free DNA sequencing assays, the sensitivity is really dependent on the recovery of short fragments, which are related to the fragment length profile of microbial cell-free DNA. And so one really easy strategy is just to change the DNA isolation strategy you use. So to use one that is more sensitive to short fragments. And finally, there are new ways that we're proposing that we can combat the effects of environmental DNA contamination. And so the method we introduced uses bisulfite tagging to identify and remove contaminant DNA. And at the same time, this allows us to perform methylation profiling, which will allow us to separate infection from disease. So I've shared a lot of work today, and it wouldn't have been possible without, of course, the help of my lab and our funding sources. I covered a number of papers from our lab as well, and if you're interested in learning more, you can look at these QR codes to get links to the paper. Thank you.
Thank you so much, Ms. Chang, for a really fantastic presentation and such a comprehensive showcase of the challenges in liquid biopsy and all the innovative technologies that you're developing at your lab. I see that there is a question already from Meredith Carpenter at Bioscience. She's asking, could you expand on the bioinformatics analysis pipeline being used during cfDNA and other studies?
Sure. So I'll first point you to this plot here. How these preparations work is very similar to standard sequencing. So in the first step, of course, we want to remove the adapter sequences. We then align all the resulting reads to the host genome. Those that align to the host genome, we save to look at the tissue of origin. And we use this by mapping to different reference methylation profiles. And I'll get into that a little bit more in a second. But for reads that don't align to the host genome, we can then align that to a database of microbial sequences. So for example, using BLAST and the NCBI reference RepSeq genomes to then quantify the relative abundance of different microbes. For bisulfite treatment in particular, we can look here. And again, we have host read removal is the first step. After that, we want to look at the read level. And so for cfDNA in particular, we're really concerned with the conversion of molecules because this indicates both how well our conversion worked, as well as if we're able to identify if it's a contaminant. And so for here, we look at different areas that are either C-poor or have, we look at the coefficients of variance across the whole genome. And from there, we can use different methods to set thresholds and figure out whether or not a molecule should be identified as a contaminant read or not. And our paper goes a lot more into depth into the actual tools that we use. So I recommend that if you're interested, you can look there to look at the exact tools and parameters we're using.
Thank you, Adrienne. I actually had a couple of questions regarding the cfDNA technology. Given that bisulfite treatment can be pretty harsh, have you looked at other modifications? So especially, if you're concerned about retaining the fragment length information, because that's something that we are interested in too. Have you looked at other modifications to instead of using bisulfite?
Yeah, we have started looking into a few other ones, such as tagging with biotin beads. One major problem and one major thing that we've done in this is we're actually tagging directly in urine and plasma. which is actually surprisingly difficult. For example, in plasma, there's a lot of proteins and other things that will coagulate and make it actually difficult to target the DNA. But we are looking into different ways in which we're actually not modifying the DNA, because while bisulfite conversion, of course, does allow us to perform methylation profiling, it significantly reduces the search space because, of course, now we only have 3 nucleotides that we can use instead of 4.
Thank you. So the other question I had was on your the urinary tract infection analysis that you did. Have you looked at the profiles in case of UTIs that occur independent of kidney transplant? Have you seen if the microbial profiles are any different?
We have not. We have looked at individuals that had stents removed and looked at their microbial profiles before and after, and we are not seeing any difference, and we're not really detecting any significant species that stand out.
If there are no more questions from the audience, please join me in thanking Ms. Adrienne Chang for a fantastic talk. Thank you so much for sparing time and sharing this amazing research that's being conducted. We have recorded this presentation, so once we format it, we will make this available on our website, and we will email a link to this video too. And in that email, we'll share our contact information in case the attendees have additional questions for the speaker or for Claret Bio. Once again, I would like to thank everyone for joining, and I hope you will attend future seminars that we would organize. Thanks once again, and have a nice day.